Race car. Look, I have apps. You have apps? You can take hold on and look.
Ready to get started? Sure you are. Sure you are. So did y'all have a good Thanksgiving? Are you all refreshed? You ready for the last sprint? No? 1 week. 1 week. I will remind you tomorrow, but Wednesday, please bring your laptop to lab so you can do student evaluation. Okay. We'll do that right at the beginning of the period, so that's out of the way. Yes, ma'am.
Can you just give a reminder, is everything pretty much due Friday?
No, everything is not pretty much due Friday.
Okay,
The food and urine exercise is due Friday.
okay.
Okay, and remember you have lab period Wednesday and lab period Friday to do what you need to do. Okay. Final, when is final? Tuesday at 8. Okay, Tuesday at 8 a.m. And then the unknown is due by end of day on Tuesday. Okay. And remember, really, the instructions for the unknown, what you have to turn in, are in exercise number 3. But basically, it is your PowerPoint. And I think in the instructions I said originally you have a PowerPoint of the pictures and then an Excel spreadsheet of the results, but then later I said, why don't you just put your results on your PowerPoint pictures? So really I need a PowerPoint of your results, and then there's some things that you have to do in terms of your write-up, including, you know, telling which keys you used and the steps you used in the keys, and then very importantly too, a comparison of your results compared to the published genus descriptions in Berge 7th, and then the comparison of your results to the published species descriptions in Berge 7th. Okay? And you don't have to have results for all of the stuff in Berge 7th, okay? There's a bunch of things that are at the species level, tests we haven't done, okay? But if it's a test you use particularly in the keying process, or one we did in class, then I would like you to address whether it agrees with that description or not. Can you have discrepancies at the species level between what Berge says and what your results are? Absolutely. Okay. These bacteria do mutate. Okay. And some of these tests too, as you look at them, you see that some of the results are a little iffy and kind of open to interpretation. So I would expect probably that you would have some minor discrepancies. Will you have discrepancies at the genus level? It's possible, but I would hope that you would see less discrepancies at the bigger genus level than you would at the species level. Another question that comes up a lot: if I miss it, do I fail the unknown exercise? And the answer is absolutely not. Okay. A bulk of the points come from reporting all of your results, having all of your pictures, and going through the process. I have had people who have gotten the quote-unquote wrong answer, meaning they reported a different species that I gave to them, and yet they still have gotten the majority of the points for the exercise. Why? Because they had all their pictures, they had all the results, they did all their comparisons, they looked through multiple keys. What sometimes happens is if you have one atypical test, and that happens to be a very important test in the keying process, you could get thrown off into a different section of the key. Okay? The thing that will kind of alert you to that, though, is if you start running against a bunch of genus discrepancies. So, for example, you use that key C1 where it says your unknown's guaranteed to be in here. Okay? And you follow it through and you say, yeah, that's my genus. And you start looking at it and go, wow, that doesn't match, that doesn't match, that doesn't match. You know, I mean, getting a lot of discrepancies. Then you're going to kind of want to go back to that key and you're going to kind of go, wow, were there any of those tests that we said were a little iffy, or the results were a little bit, you know, we were unsure about? Then you can kind of go back and look at your pictures. In some cases, we can repeat some tests. But if you have inconsistencies, that's a possibility. That's why I ask you to do keying using more than one key. If you can get the same answer for the same genus by multiple methods, then you're much more confident in your results. Okay? But don't put all this, pressure on yourself. Is if I get it wrong, then I get 0 out of 150. No, that's not true. So the report is not lengthy. I mean, the time that it takes kind of is to take your pictures, get your PowerPoint organized and things like that, but it's not a traditional lab write-up with materials and methods, introduction, et cetera, et cetera. The amount of material you have to report is very well defined, and just make sure you hit all the bases, all the things I ask you to do, make sure you do. Natalie did catch up with lecture recordings this morning, so they're there. I did find last year's exam, number 4, which I hadn't posted, but it doesn't have answers on it, so that's I guess, for you to figure out. But if you want to see what the questions were like last year, I posted that. Study guides are up to date. And then I sent out, and it's now on the website too, those of you that are madly looking for optional points, there's another set of activities, the videos.
How
So,
long are the videos per video? Probably gonna kill us,
I mean, half an hour maybe per video. It's been a while since I've seen one. So, anything else? Okay, last topic. We're going to talk about some applications of microbiology now. And these applications are particularly related to what we've just learned about the immune system. So we're going to be talking about a little clinical microbiology. This is what John intends to be doing next year. Those of you that know Kate Miller, this is what Kate is doing right now. The purpose of clinical microbiology is to use tools to try to identify the presence of disease. Historically, these particular tools have been referred to as serology or serological tools. What's the derivation of that term? What kind of tools are we talking about? What kind of techniques are we talking about if we're talking about serology or serological techniques? Yeah, looking at the blood. Okay, blood analysis. Okay. And when we do these serological tests, we're really looking for, in general, one of two things. Can we detect the presence of pathogens? Sometimes it is not convenient or even possible to actually directly detect the presence of the pathogen itself. So many of these serological tests are antibody-based. And instead of looking for the pathogen itself directly, we are looking for specific antibodies that have been produced in response to that infectious agent. So if we see Treponema pallidum, that's a caustic agent of syphilis, antibodies, then we can infer that you either are currently infected or were infected with that pathogen at some point in time, and therefore the presence of these antibodies infers the existence of the infectious agent. Now, all of these serological tests are based upon the presence of antibody-antigen reactions. And one of the things that is crucial about these reactions is they are highly specific. So a particular antigen induces the formation of a particular antigen. And you now know why that specificity exists, because of the way that the B lymphocytes work and how they are activated by particular antigens interacting with their receptors, and that those B lymphocytes then produce a single type of antibody. Now, one of the things that we do have to worry about in some of these serological tests is the possibility for cross-reactivity occurring. And that is that a particular antibody may react with more than one antigen from a different source. We talked about that when we talked about types of antigens. And the fact that in different species, these heterophile antigens, a group A streptococcus antigen and a heart valve antigen, can both react with or cause the formation of the same antibody. So in these serological tests, we really want to make sure that we include the proper controls. And if we do so, then we can eliminate, very likely, any false positive tests that might result. Okay? You don't want a false positive in these tests. You don't want to go and collect a blood sample and say to a person, you're suffering from HIV, when you aren't. Because the regimen of treatment for HIV, for example, has its consequences on the body. And so you want to make sure that you are very confident that the results of these tests are accurate. Very likely, particularly in the case of serious disease, you don't rely on the results of a single test. So in the case of HIV, if it's initially diagnosed by one testing mechanism, then usually there are secondary tests that are done to confirm that initial positive test. So we're going to give you just kind of an idea of some general kinds of serological tests and how they work and what their basic function is. Now, in recent years, serology has really expanded so that we don't just have to test blood anymore. There are serological tests that have been developed to use cerebrospinal fluid. So you take a spinal tap and you can do some of those same serological tests that you did with blood. Serological tests have been used to test saliva. And a fair number of urine-based serological tests are used as well. What's probably the most famous or widely known urine-based serological test? Drug test, but that's testing for presence of drugs. Pregnancy test tube, yeah. In that particular serological test, what are we testing for? Hormones. HCG, the human chorionic gonadotropin hormone, the pregnancy hormone. And there are antibody-based tests to test for the presence of that hormone. And they've gotten increasingly more and more sensitive over time, so we can really, in these serological tests now, detect pregnancy very early on. Relatively small numbers of these molecules of HCG can now be detected. So what we'll do first is we'll kind of do a little review, because it's kind of in parallel with what you're doing with your unknown right now, but talk a little bit first about how we can test directly for the presence of pathogen. So what are some kind of tests that we do for pathogen detection? And then we'll spend more time talking about the antibody-based testing and how we can test for antibodies that were produced by that pathogen. So here are some typical pathogen identification methods. Three basic categories: phenotypic, genotypic, and then the immunological. Okay? So phenotypic involves mainly microscopic observation of characteristics. It also includes physiological/biochemical characteristics, and in some case, some chemical analysis. So, this is your unknown identification, basically, in a nutshell. Okay? We started out first by looking at microscopic morphology. A lot of staining characteristics, size of your unknown. Does it produce endospores? What's the Gram reaction, etc.? Does it have pH-feed ring? Is this an acid fast? Some additional information that complements that is macroscopic morphology, particularly of colonies. So you need to learn those terms associated with margins and elevation and form. Okay? The early editions of Berge's manual, particularly up to the 7th edition, relied primarily on those morphological characteristics. In the 7th now, there's a fair amount of physiological information that's been added to the identification scheme. All of the metabolic tests that we did in the second part of the semester. Do you ferment certain carbohydrates? Are you oxidase positive or negative? And in the 7th edition, but into the 8th edition as well, a little bit of immunological testing started creeping in. And I don't know if you've started working on your unknown yet or not, but in terms of the species scheme, particularly in the 8th edition, when you start getting into some of the genus of gram-negative bacteria, like Salmonella, you'll see a huge amount of information about Salmonella serotypes. This is an attempt to try to drill down on more finely detailed analysis of different kinds of Salmonella, using immunological testing to look for the presence of particular antigens on particular bacteria. A lot of these antigens are part of flagellum, for example. And so they really kind of went almost overboard, I think, in the 8th edition in Salmonella, breaking it down into all of these serotypes, adding purified antibodies to those particular bacteria, and then seeing if the antibody reacted with it or did not. If the antibody reacts with it, then these particular antigens are on the surface of these particular cells. So that really gave a lot of detailed information. Haven't done much we haven't done any chemical analysis, but there are chemical analysis techniques that are used to identify specific chemical composition. Certain cell wall peptides, certain cell membrane lipids. So, here's a very simple representation of some phenotypic methods, an immediate direct examination, gram stain. What we're going to see, we're not going to talk about it right now, is that some of these direct microscopic methods are enhanced by the use of immunological techniques. So for example, we will talk about some fluorescent antibodies that can be used in a type of direct observation called fluorescence microscopy. And it really does provide a whole picture of the organism that you can't see now with just typical bright field microscopy, which is what we use. So the flow for typical phenotypic method is cultivation of the specimen, and then looking at colonial appearance, growth requirements, biochemical testing, etc. This can be a difficulty, cultivation of a specimen. When we talked about the diversity of microorganisms, I told you that microbiologists used to think that the ocean was a very poor environment for microbial life, and that there really wasn't much diversity of microorganisms in the ocean. That's because we can't culture them in the lab. Do you remember how we then determined the presence of all of these species without being able to grow them?
There was residual DNA that's been in water.
Yeah. I mean, there was DNA, even not residual, from the organisms, and by doing various sequencing methods, we could identify novel DNA sequences and RNA sequences that then inferred the existence of new species of microorganisms. So these genotypic methods are becoming increasingly used to identify microorganisms. They look at the genetic makeup rather than the product of the makeup. That's important if you think about it for a second. Because really, isn't colonial morphologies and things the result of genes? Isn't the presence of a particular enzyme, which means you can ferment a sugar or not, the product of the gene? Yeah. So this really is getting us looking directly at the genes. It's very precise. There are automated methods that have been developed to do this genotypic testing for identification of species, and you can get very quick results. Some of the genotypic methods that are used involve DNA analysis and hybridization. You all familiar with the concept of hybridization? In hybridization, basically, what you take is some little known DNA sequence that has been labeled some way, maybe with a chemiluminescent drug, maybe with radioactivity, and then you incubate that with unknown DNA sequences. And if the two sequences are complementary, if the probe is complementary to the sequence you're trying to identify, then they will bond together by hydrogen bonding. They will hybridize. And then there are techniques you can use to detect the presence of these hybrids. And one of them is electrophoresis. So you can run these electrophoretic gels and see if hybridization has taken place. Another technique that's increasingly being used in genotypic detection is PCR. We're going to be more interested, though, since we're spending a whole lab basically doing identification of organisms, to look at the other side of things, to look at immunological methods, which involves the use of specific antibodies to detect an antigen. Sometimes it's easier than testing for the microbe itself, particularly if it's difficult to culture that microbe, and you don't have access to the high-tech, expensive methods of genotypic testing, because that's not cheap. These immunological-based methods are much more cost-effective, and they don't require the level of expertise, typically, that it does to run these more complicated, sophisticated machines. But again, these immunological methods, to be useful, have to be highly specific, and they also have to be as sensitive as possible. We're going to talk about some highly sensitive methods of immunological detection, which theoretically are sensitive enough to detect the presence of one molecule of antigen in a sample. That's incredibly sensitive. That's one of the things we've seen in the evolution of the development of these techniques is, over time, methods have been developed to make increasingly sensitive testing mechanisms. Some of these methods that I'm going to show you are a little bit old and outdated and really aren't used that much anymore. But they provide kind of a general understanding of the basis of many of the newer methods. Another reason I like to show some of these is to give you an idea of how creative some of these methods are, and how with a limited amount of technology you can get some very powerful information if you are clever enough in devising a particular test. So, serological methods have traditionally involved looking at blood, but now include other fluids as well. Serological methods involve the formation of visible reactions that allow you to test for the presence of this antibody-antigen complex that's forming. So you've got to be able to detect whether this complex is formed or not. So if you have a purified antibody for a particular disease, and you incubate that with blood serum, and there's no complex formed, we know the antibody was there, we added it. So the antigen must not have been there. That would be a negative result. But we've got to be able to detect whether this has happened or not. And sometimes that's tricky. Sometimes that antigen-antibody complex is directly detectable. And other times we have to do some interesting things to be able to see whether this reaction has occurred or not. Yeah.
So, just to clarify, I was thinking, I guess, that they were testing for the presence of antibodies, but instead it's that they're adding antibodies and testing for that complex.
Depends on the test and what you're testing for, okay? In some cases, you really want to know whether the antigen's present. So in that particular case, you would take a blood sample where you wanted to know if the antigen was present and add purified antibody to that. In some cases, you want to know whether the antibody is present. So then you would add known antigen to the blood sample and see if it reacted. Okay? And we'll give you a concrete example in just a second of that. Another thing that's important to understand about these is that typically these serological tests don't give you concentrations as results, but instead they will be expressed in terms of titer. We already talked about that concept of titer earlier on. Titer is kind of a relative term. It's kind of an order of magnitude comparison. Does this sample have 10 times the antibody this other one does? Does it have 100 times? Does it have a tenth of the amount? So titer is kind of a relative comparative level. So let's give you a classic example of when you might want to know whether your blood has a particular antibody. Anybody know what rubella is? Measles. Okay. Measles used to be a fairly common childhood disease until we started immunizing. Measles is particularly devastating to fetus, particularly in their first trimester. So if you are a woman contemplating becoming pregnant, you really should know what your rubella titer is, what your antibody level is against that particular pathogen. If your titer is high, then if you are exposed to measles, your antibodies that you formed previously will react with that and eliminate the threat before it can infect you, but more importantly, before it can infect your child in utero. So you trot off to the doctor and get a blood sample taken. What do? They isolate the blood serum, and then they incubate your blood serum with known rubella antigen and look for the formation of those rubella antigen, your antibody complexes form. And the more you can dilute your serum and still get the formation of this reaction, the higher your rubella titer will be. And what if your titer is deemed to be insufficient? You're not really carrying around enough antibody protection. What are you going to do? Get immunized, okay? But it's really important to know this because of the effect that this particular disease has on your fetus. Okay? So again, in some cases, we are looking for the presence of a particular antibody in your serum, in the case of rubella. So we take some patient serum, antibody content unknown. We don't know if there's rubella antibody or how much. We have purified antigen, and then we look for the presence, when we mix those two together, of an antigen-antibody complex. Okay. In other cases, we really want to identify the organism itself. So we might have some unknown organism, but we want to know what it is. So we take a sample of that, maybe a colony, and then we take here antibody of known identity. Maybe that's a specific antibody for a specific flagellar protein. And then we mix those together. And we look for the presence of that antigen-antibody complex. And if it's present, then we know that particular antigen is present because it reacted with the antibody that we knew we added to that particular sample. Okay. So those are the two kind of different types of serological testing. Testing the blood for antibodies or testing pathogens for particular antigens being present. One of the oldest serological tests is human blood typing. How many of you know your blood type? Blood type. Next time you're at the doctor, ask what your blood type is. Okay? Just good to know. If you are blood type A, what does that mean in terms of antigens on your red cells and antibodies that you might have in your serum?
You have antibodies against like all the.
Okay. Yes, you do. Let's take the red blood cells first. If you're type A, what antigens do you have? You have A antigens. Okay. And remember that there are a pair of alleles. And so if you are type A, which is a phenotype, what are your possible genotypes for type A? Okay, I is the recessive allele. Okay. Should you have type A antibody circulating around in your blood? Hopefully not, right? Because what would happen with your A antibody? Yeah, it would bind with these A's right here on the surface of your red blood cells and coagulate, or a more technical term for this is agglutinate. But you do have B antibody naturally floating around in your blood. It's not reacting with your cells. So, by taking your blood and then adding known antibody to that and seeing whether you get an agglutination reaction or not, you can determine your blood type. And you can do that for the AB antigens, and you can also do that for the RH antigen. So, one of the ways that you can do a direct observation of whether this antigen-antibody complex is formed is to add antibody to antigen and see if agglutination occurs. Agglutination is basically a clumping. And this typically involves pretty large antigens. Oftentimes, these antigens are actually the part of a cell surface. And remember that antibodies have a valence of at least 2. IgG has a valence of 2. Remember what the valence of IgM is? 10. 5 IgG-like molecules in that IgM. So remember that the fact that these have a valence of at least 2 means an antibody can bind to more than one antigen simultaneously. And it can cross-link them. And that cross-linking that occurs between these cell surface antigens results in a clumping of those antigens. And that is oftentimes visible in the form of this clumping reaction you can visibly see here in an agglutination reaction. And you can get some level of determination of how much antigen is present by doing a serial dilution of the serum and then finding out in what dilution is that clumping occurring. So the more you can dilute it and still get a reaction, the greater the titer will be. One of the first tests that were developed for using agglutination reaction for the identification of disease agents was Rocky Mountain spotted fever. Anybody remember way back when we were doing a survey of the microorganisms, what type of pathogen causes Rocky Mountain fever? Rickettsia. Okay. Little tiny small bacteria that they once thought were virus. Sometimes the agglutination reaction doesn't happen very well if the antigens are getting too small or the cells are too small. So sometimes they will take the pathogen and they'll actually cover the surface of little tiny latex beads with the pathogen. And then they will mix the antibody there. And those latex beads help to give you a better agglutination reaction that is much easier to see. So this is kind of a test where you can just really have a slide and you can just kind of see this clumping occurring. So it's actually a visual test. The Weidel test, which was a test for the presence of salmonella, was developed quite early on. That is another agglutination reaction. There's a agglutination test for walking pneumonia called the cold agglutinin test. This is kind of older technology, but it really is important still in testing of some particular pathogens. There's a variation on this that has the same principle, called a precipitation test. This one is typically used to test for the presence of smaller, more soluble antigens. So the antigens don't have to be part of the cell surface. Oh, forgot. Before I do the precipitation, let's do this one. This is kind of a creative test. A pretty creative method was developed called the viral hemagglutinin test. And this was originally developed for testing for measles, mumps, influenza. I think there's one more. This particular test is based upon a competition reaction that occurs between human antibodies and hemagglutinins, which are basically red blood cells. The thing that's cool about this particular test is that these particular tests can be done in little microtiter plates. Have you all seen those plates before, the little plastic plates with all those little wells in them? You can do 128 tests at a time with these particular tests. And it does result in a visible reaction. And the visible reaction that you're trying to detect is, has hemagglutination occurred? Have red blood cells clumped together or not? And the way you determine that basically is you look at the well in the microtiter plate and you determine whether those red blood cells that are part of the test have floated to the top of the well or have sunk down to the bottom of the well. So if you look at this particular microtiter plate here, this has got several different patients that we're testing. In this particular case, this is a measles test. And we've got various dilutions of that patient's serum. And then we've got room over here for negative controls and positive controls for each serum. Okay, this is what it looks like when hemagglutination does not occur. The red blood cells have not clumped together, and therefore they sink to the bottom of the microtiter plate. And they look like kind of a little button down at the bottom. This is what it looks like when hemagglutination has occurred. And the red blood cells have basically linked to one another and have formed this lattice that actually floats. It's kind of like a little raft almost. Okay? So here's hemagglutination. That's a negative result, as it turns out. We'll see in a minute. So in that particular case, the red blood cells float at the top. And here is lack of hemagglutination, so the red blood cells sink to the bottom. Okay, so here's our test. Red blood cells, which are the hemagglutinins. If measles virus are present in the sample, then those measles virus bind to antigens on the surface of their red blood cells and hemagglutinate the red blood cells. Down here, what if there are antibodies present against that virus? Then those antibodies will bind to the virus. We add this known virus to the red blood cells. And if antibodies are present in the patient's serum, they will bind and they will agglutinate the virus. And the virus, therefore, will not be available to agglutinate the red blood cells. So the red blood cells, not agglutinating, will sink to the bottom. So in this particular case, this would be a negative result for patient antibody against measles. You have no measles antibody if this happens, because there's nothing to compete with the binding of the virus to the red blood cells. The red blood cells bind, hemagglutination occurs, floating occurs. But down here we have the measles virus, and it binds the source of measles antibody. It binds to the virus. So now agglutination of the red blood cells can occur. It sinks. So in this particular sample of results, which patients have some level of antibody against measles virus? In these samples right here, which individual patients, A through H, have measles antibody? B, C, F, D, F, G, H, right? Which one has the higher measles titer, antibody titer? F has, because we can do more dilution and still get that reaction occurring. Okay? So you can look directly at these plates and you can go, wow, I can tell which people have measles antibody, and I can get a titer too. And this can be automated because this can be run through a plate reader, and the plate reader will read those results because you'll get different absorbance when you have the solutions that are agglutinated and not. Okay. So this is a situation where technology has taken the basic premise of an agglutination reaction, but spiced it up a little bit so we can very conveniently get multiple patient samples tested, and we can also then get titers for that at the same time. So if I want to modify this test to test for HIV antibody, okay, how do I have to modify the test? I just have to add to the serum and the red blood cells a different antigen, in this particular case HIV antigen. Okay. This particular test can be modified a number of ways. Okay, we will keep going tomorrow and talk about some other serological tests.